Okay, so adaptive immunity, I'm going to go on ahead and I think I'm going to split it into 4 different parts. Because of how it's set up, we kind of have to go through the background of how everything works first. Then we're going to go through the 3 primary cell types in adaptive immunity: the helper T cell, the B cell, and the cytotoxic T cell. So what we're talking about right now is going to be this side over here. We're going to be talking about the adaptive immunity and how all of this works, at least at first. And then we're going to talk about the helper T cell. Then we're going to talk about the B cell, which is the antibody mediated immunity portion of this, and the cytotoxic T cell after that, which is the cell mediated immunity part of this. So let's talk about, and I know I say so and now a lot. I've realized that since I've started recording. Please forgive me. I'm sorry. I think I say it in the classroom too. It just doesn't seem so obvious to me when I do it in the classroom. Let's see. Okay. How do we make lymphocytes? So the 2 types of lymphocytes that we primarily are going to discuss are going to be the B cells and the T cells. Both of them originate in the red bone marrow. Part of this we've kind of already talked about when we were talking about the general lymphatic system. But B cells mature in the bone marrow, and T cells, while made in the bone marrow, actually have to mature in the thymus. The thing about B and T cells to keep in mind, the thing that you have to remember about them, once they hone in on a target, they kill that target. So you kind of have to have a checks and balance system for them. You have to make sure that while they are doing that, they're also not going to kill you. So there are 2 checkpoints for both of these cells. The first checkpoint is called positive selection. When we talked about the red blood cells way back when, we talked about the antigens, those proteins that are on the surface of a cell. Your cells have self-antigens, basically identifying them as belonging to your body. So when they get checked, your body goes, yeah, these are okay, they're self-antigens, I recognize this, I don't have to kill it. When something has a foreign antigen, however, is when your body needs to kill it. So the first step is, let's make sure that these cells recognize antigens. So that's positive selection. Do they work? If they work, great, they go to the next test. If they don't, at this point, I kill them. There's no reason to keep something around that doesn't do a job in my body. It's just wasting energy for me. So if they pass the first test, they move to the second test. The second test is negative selection. Now, remember what I said. They hone in on a target and then kill that target. I need to make sure that whatever target they recognize isn't me. If they hone in on me, they will kill my cells too. So negative selection is basically making sure that they don't recognize me. In other words, if they respond to a self-antigen, I kill them because I don't want them attacking my own body. Autoimmune diseases is when this step goes wrong. When your immune system is attacking your own body, somewhere in the scheme of things, when testing was done, they got past negative selection. So if I pass positive selection and negative selection, I get to move on into the lymphatic tissues and into general circulation. If I don't, I either die here or I die here. But either way, I have to pass both tests before I'm allowed out. The primary lymphatic organs are the thymus and the bone marrow. Primary meaning this is where the cells get made. Remember, the thymus makes the T cells, the bone marrow makes the B cells. The secondary lymphatic organs are where those cells work. So the tonsils, the spleen, the thymus, the lymph nodes, all of these organs are where they actually function, where they live their day-to-day lives, not where they began. There are two major types of adaptive immune response: cell-mediated immunity and antibody-mediated immunity. Okay? And like I said, this first lecture is kind of background. Cell-mediated immunity is used when viruses or bacteria are hiding inside of the cells of your body. In other words, they're not just free floating around, they're actually hiding inside of your cells. So it recognizes infected cells. Who is responsible for cell-mediated immunity? Cytotoxic T cells are. Okay? Cell-mediated immunity. It's recognizing infected cells, cells that literally the virus or the bacteria is hiding inside. And cytotoxic T cells are this division of immunity. They're the ones in charge of it. Some of these cells can go out and attack. Some of these instigate an immune response. Okay? It just depends on what the situation is. But cell-mediated immunity is cytotoxic T cell all the way. That's who's in charge of that. Antibody-mediated immunity. We also call this humoral immunity. Way back in the day, any liquid that was found in the blood any liquid that was found in the body was called a humor. Whether it was cerebrospinal fluid or blood or lymph or it didn't matter. They just called it the humors of the body. Don't ask, I don't know. But because of that, they also call this humoral immunity. Okay, remember that in cell-mediated immunity, the virus or the bacteria had to be hiding in one of our cells, right? In antibody-mediated immunity, these are invaders that are in the body fluids. In other words, it's the actual virus. It's the actual bacteria. It's the actual toxin. It's not hiding inside a cell anymore. They're just exposed on the surface somewhere. Okay? B cells are responsible for antibody-mediated immunity. So just like cell-mediated immunity with cytotoxic T cells, B cells are responsible for this. Why? For all intents and purposes, B cells are antibody factories. So them being in charge of antibody-mediated immunity makes sense. They release antibodies into the fluids of the body, and antibodies are like a homing beacon. A homing beacon is something that you attach to something else so that somebody can find it. Think about the app for parents on their kids' phones. They put it on the phone so that if they can't find their kid, they can actually locate their kid by honing in on where their phone is. It's something like that. So, the antibodies are basically those that mark who needs to be assassinated. Okay? Antibodies are a blinking red light to the phagocytes, for example. The phagocytes eat who needs to be killed. Yeah, this is the target that I put on the back of that bacteria and go, hey, that guy, do you see him? You need to kill him. So we've got two major divisions to adaptive immune response. Cell-mediated immunity, which is going after those things that are hiding inside of our cells, and it's controlled by cytotoxic T cells. And we've got antibody-mediated immunity that's controlled by the B cells that release antibodies into the fluids of the body that go and attach to exposed viruses, exposed bacteria, exposed toxins, and basically mark it for death. And it's controlled by B cells. I don't remember if I said that. Okay, so all of this stuff, and yeah, we recognize something, we recognize a target. Let's talk about exactly how it is that we do that, because there has to be some process so that we know I'm going after cold 2B. Remember I said little Susie comes home with cold 2B. If she came home with cold 2A, the 2B stuff wouldn't work. How does that function? How does that work? Okay. Lymphocyte activation. So remember, our lymphocytes are our B cells and our T cells. You recognize and respond to and remember a particular antigen or haptens, which is basically chunk of antigen. An antigen is a protein displayed on the surface of a cell that can produce an immune response. We have two different types of antigens. We have self-antigens. Remember, I drew that cell and I said I've got a protein that says Ronica, so it's a Ronica protein. So if my immune system comes along and sees that, it goes, oh, that's a Ronica protein. That's perfectly normal. No big deal. That's a self-antigen, and it will not elicit an immune response. Okay, so this will not cause your immune system to react to it. Okay, your immune system doesn't react because it goes, oh yeah, that's normal. That looks normal. A haptens is a partial antigen, okay? It has to be linked to a carrier protein to elicit an immune response. Hang on. You have to remember that these proteins are not little teeny tiny things. They're big things that have a whole bunch of parts and pieces. I don't have to recognize the whole thing. I can recognize one part of it and it's still enough for me to recognize. So, sorry guys. I literally just choked on nothing. So let's say that I've got a cell. Well, actually, before we even get into that, I just talked about self-antigens. Let's talk about foreign antigens. Foreign means it doesn't belong there, and your body knows it doesn't belong there. So for a self-antigen, I had Ronica, right? Well, let's say for a foreign antigen, I have Monica. Okay, this is the foreign antigen. And you'll notice that, let me redraw that because that's really in a bad spot. You can't even see it. Monica. Let's say for argument's sake, okay, that these are each letter is a part of the antigen. Now, I have an O, an N, an I, and an A in my name. So O, N, I, and A. What I don't have is an M, and I don't have a C. So remember I said it doesn't have to be the whole thing. It can just be a part that it recognizes. My body sees that or sees that C and goes, that's not normal. That doesn't look like my normal self-antigen. My normal self-antigen has an R and it has a K. This doesn't make sense. So when it decides that there's something wrong with this and it decides to attack this, it actually recognizes, it understands that this and this are wrong. So what it recognizes as being foreign are the parts that don't look normal. So self-antigen looks normal. Foreign antigen, not so much. And it's the foreign antigen that's going to basically raise the alarm that there's something wrong. So, lymphocyte activation. There are two general principles to lymphocyte activation. One, the lymphocyte has to be able to recognize the foreign antigen. This is the positive, yeah, my brain's not working, sorry. The positive selection. Remember I said it had to recognize that something was there. So, one, it has to recognize that there's a foreign antigen. Two, after it recognizes it, it has to become activated. if I have one cell that recognizes the invader and I have 5 billion invaders, 5 billion bacteria in my 400 trillion cell body, one cell, that's like saying I've got an army of 1,000,000 and I've got one soldier on the other side. Yeah, that's not going to work. So what do we have to do? I have to increase the number. So once I understand that I recognize this foreign invader, I have to build an army, okay? I have to get my numbers big enough that I can actually go and destroy this enemy. Antigenic determinant. This is a part of a foreign invader. So remember here, I said the M and the C were foreign. They didn't look normal. Well, in this case, this is an entire virus. Yes, I know it's a bacteriophage, which doesn't infect humans, but just go with me on it because it was the one thing I could draw that actually kind of looked like it was supposed to. If you look at all the parts and pieces over here, they are each individual parts and pieces, just like the name letters. Each part that is different that can be recognized is called an antigenic determinant, and each one can be used to identify this particular virus. Why would that be important? Well, my lymphocytes, okay, have these things on their surface. They're called antigenic receptors. The base of these are called the constant region. All of them are the same. But the tips out here vary. That's why they're called the variable region. And depending on what tip you have, it determines what you recognize. So you'll notice I've got kind of an orange tip, a blue tip, a green tip, a yellow tip, right? So if I have one lymphocyte that recognizes the yellow leg, I have another that recognizes the green leg, I have another that recognizes the blue capsid, I've actually increased the numbers in my army because now I've got a more diverse army that can recognize different targets on the same target or different parts of the same target. So for example, if I had one army that recognized the face of a villain, if I had another army that recognized their fingerprints, if I had another army that recognized their DNA sequence, I actually have multiple ways of catching this criminal. So if I've got one lymphocyte that recognizes yellow leg, another that recognizes the green leg, another that recognizes the blue capsid, it basically means they all recognize the same target, but each binds to their own antigenic determinant. So we now have this diverse population of assassins all recognizing the same target. Kind of like having a criminal and having all 10 fingerprints, each one of them being unique instead of just one to catch them. Each lymphocyte, each one of these cells has 10,000 of these receptors on its surface. Now, I've drawn this with artistic license because I'm not an artist, but they actually have 10,000 of these on their surface, basically meaning that, in all honesty, if I touch this and it touches any of these that are on my surface, I'm going to recognize it. So the receptors on the surface for each activated lymphocyte can now make copies or clones of itself, so even more can recognize the target. I'm building these armies. Now imagine more than one recognizes the target. We have even more cells that recognize and kill the invader. So how do we actually warn the immune system of an infection or an invasion? We have these things called major histocompatibility complexes. Okay? Major histocompatibility complexes, MHCs. We have two types, MHC1 and MHC2. Okay? MHC1, MHC1 is a protein inside of my cell that is found in every nucleated cell that I have in my body. If you have a nucleus, you display MHC1. There's only one not nucleated cell in our body, and that is going to be our red blood cells. So every cell in our body except those has this. And it's basically used by the cell to indicate that they are infected. Remember what I said about viruses and bacteria getting into your cell and hiding inside? This is a way for that cell that has a virus hiding inside to basically raise a flag and say there's something wrong with me. Okay? So these cells displaying foreign antigen MHC1 complexes are gonna die. Your body is very intentionally going to kill them because they are not normal cells. Who recognizes MHC1? Cytotoxic T cells do. Remember that cytotoxic T cells are the ones killing the cells that have been invaded. So this makes sense. They kill infected cells. This cell is infected and basically displaying something saying, hey, I'm infected. So the cytotoxic T cell goes and kills them. MHC2 is an entirely different ballgame. MHC2 is found in antigen presenting cells. If you're presenting something to somebody, it means that you're showing them something, right? Antigen presenting cells are basically doing the same thing. So MHC2 is used by antigen presenting cells to indicate that there is something foreign in the body and it needs to be found and killed. Hey, I found this wanted poster. Let me show it to you, and let me show it to you, and let me show it to you. I am not the killer in the poster. I don't need to die because I'm showing this poster, this wanted poster, to people. That's MHC2. The antigen presenting cells are just saying, hey, look what I found. You need to be aware that this is in the body. These cells are a call to arms. They're the ones that get the immune system to actively seek out the target. Me going around with that wanted poster is basically warning the population, hey, there's something in the body. You need to go look for this. Okay? Now, who recognizes MHC2? The helper T cell does. Okay? Notice it doesn't say cytotoxic. In other words, these cells, the antigen presenting cells, they're not going to die either. They're just the person holding the wanted poster. There's no reason for them to die. Okay? So let's talk MHC1. So here's a body cell, normal body cell making normal proteins all day long. Any normal protein that is inside this cell is going to get displayed on the MHC1 molecule. Doesn't matter what it is. If it's a protein being made in this cell, I display it on the surface and go, hey, look. And your body goes, that's a normal protein. Cool. So when it's normal and a lymphocyte comes along and checks it out and goes, yeah, that's normal, it leaves your cell alone. It goes, yeah, that's fine. No big deal. Now, mind you, this lymphocyte is a cytotoxic T cell. Why do I know that? Because remember, MHC1 is designed for the cytotoxic T cell. And as long as the cytotoxic T cell doesn't go, hey, I recognize that as a foreign protein, the cell's fine. It leaves it alone. Now, when does the issue start? Let's say that I get infected with a virus, and the virus takes over the machinery of my cell to start mass producing copies of itself. Now, I've got all of these pieces that are not normal proteins anymore. Now remember, MHC1 just picks up a protein out of the cell and goes, here, look what I found. It doesn't pick up only self proteins. So in this case, I picked up a self protein. In this case, I didn't. And in this case, I didn't. So when MHC starts displaying these things that it's finding, all of a sudden now I'm displaying something that's foreign. It's not an antigen that my body normally makes. So I've got MHC1 with a self protein, and I've got MHC1 with a foreign protein. In this case, when that cytotoxic T cell comes around and has that variable region that recognizes this target, yeah, I'm a cytotoxic T cell. There is something wrong with this cell. I need to kill it because it's not supposed to be here. Okay? So, that was MHC1. Let's talk MHC2. MHC2 is found in the antigen-presenting cells. Examples of those include B cells, macrophages, monocytes, and dendritic cells. They're used by the antigen-presenting cell to indicate there's something foreign in the body. I'm holding a wanted poster up and showing you, this guy was spotted in Alice today. This guy was spotted in Alice today. Be careful, this guy was spotted in Alice today. And let's say that you are all assassins who are, well, I shouldn't even say assassins. You are all law enforcement. So I'm showing you somebody that you need to keep an eye out for. And if you see that person, you need to kill them because they've killed 20 people, whatever. But either way, I'm showing you something that you need to see. Okay? Remember that MHC2, I'm sorry guys, that's a crap color. MHC2 is recognized by the helper T cell. Okay, so helper T cell is the one who goes, hey, MHC2, you're supposed to be what I'm supposed to be looking at. And it goes and it checks it out. If it recognizes something as, uh-oh, that's bad, this is the call to arms that, hey guys, there's something dangerous in the body. You need to see this. So, macrophage, an antigen-presenting cell, happens to come across an invading bacteria, decides he's going to eat it, eats it up, breaks it apart into pieces, into antigenic determinants, foreign antigenic determinants, and it displays them on MHC2. Notice that even in the picture, MHC2 looks different than MHC1. There's a reason for that, because it is different. We only find these in antigen-presenting cells. Okay, so this is my macrophage, displaying something that it found, the wanted poster, on its MHC2. Now, who recognizes MHC2? Helper T cells. Okay, so helper T cells come along and they go, hey, I recognize this. It recognizes it. And because it recognizes this, this guy gets activated. Remember I told you I have to build my army, right? The antigen-presenting cell is responsible for finding and activating the helper T cell that corresponds to the correct foreign antigen. Notice, my variable region here matches that foreign antigen. It has to be the right helper T cell recognizing the right foreign antigen. If I had, let's say the helper T cell had a pink variable region to it, it wouldn't recognize the green triangle, which would mean that it would just go off and go, yeah, I don't recognize that. But since this one has them matching into it, this one is activated and it starts making copies of itself. Okay? This is the rallying flag for the helper T cell. This is the helper T cell getting told, hey, you're the one who recognizes this. You need to make an army right now. And you'll see why in just a minute. So again, MHC1. can be used by any nucleated cell in the body. It takes any protein found inside that cell and displays it on the surface, whether it is a self-protein or a foreign protein. It doesn't matter. Because, or if it displays something foreign, it identifies the cell as being infected. It is wrong. That shouldn't be there. When it does display, that should be 1, MHC1, and it's foreign antigen, it's a death sentence. I am telling you that I am full of virus. You need to kill me. And the cytotoxic T cell is the one that comes in and kills this infected cell. Because cytotoxic T cells are the ones that recognize MHC1. Again, these are my notes, so T sub C is the cytotoxic T cell. T sub H is the helper T cell. MHC2 is used by antigen-presenting cells, monocytes and B cells being examples. The displaying MHC2 with foreign antigen is a warning. It's the person holding the wanted poster. It's not a death sentence for that cell. That cell isn't infected with anything. It just found something and wants to show you. Who responds to MHC2? The helper T cell does. It's the one that complexes with it. So that's going to be the history of that, okay? Now we're going to talk about the individual types of cells in the next 3 lectures.